Good morning. I'm Dr. Malone. I'm a movement disorder neurologist. And if you can't hear me, just raise your hand. And I guess online, hopefully everything should be coming through okay. And Megan's on top of that. So my topic today will be genetics in Parkinson's. And I think they just said, hit the green light. Got it. Thank you. All right. So why do we even have a Parkinson's clinic? And why do we talk about Parkinson's disease? And why is it near and dear to everybody's heart? Right? So 75% of patients in our clinics with Parkinsonism, so that means slowness, stiffness, and tremor, right? So 75% of those people have Parkinson's disease. So it's the vast majority of what we see. And genetic causes of Parkinson's make up about 30% of familial cases. And the important thing I think to note here is that I think we're talking about 3 to 5% being sporadic cases. And sporadic means random, right? So there are some hidden genetics that you may not know about. So, talking about genetic makeup of Parkinson's, there are 28 distinct chromosomal regions, and 6 of those regions, there is one gene mutation. A single gene is responsible. So most Parkinson's disease is probably multifactorial, meaning you may have genetic makeup, you may have some sort of susceptibility, whether you were exposed to something medicinally, whether you're exposed to something from a viral standpoint, and then the environment. Thinking about well water may be a risk. We know Agent Orange is certainly a risk. There are other things that cause people to have Parkinson's or Parkinsonian syndromes. All right, so basically talking about Genetics 101 today. So realizing that most people in the room were probably not biology majors and probably don't love the Punnett square as much as we do, but talking about these 4 different types will be important today. So autosomal dominant, we'll break that down, autosomal recessive, X-linked recessive, and then mitochondrial. So those are sort of the 4 basic principles if you think about genetics. And autosomal dominant and recessive you've probably heard about. X-linked recessive as well. Mitochondrial, I would say, is sort of a little bit more tricky. All right, this is your indication to think, wait, what did she just say? Have a glass of water, have some coffee, and let's refocus here. So there is a typo on this slide, so I want to point that out first. So this one is going to be homozygous. So you're looking at your Punnett square. You've got someone who has the recessive gene for blue eyes and somebody that has the dominant gene for brown eyes, right? And then in the middle of this square are the offspring. Okay, so the children are in that square. And so you'll see the dominant trait will win over the recessive trait in this particular square. All right, so what do you need to know? So if you're talking about autosomal dominant, right, you're talking about one copy of the gene from one parent can cause the condition. 50% inheritance by the child. If you switch over to autosomal recessive, it's a recessive gene, so it's 25% inheritance. So the child does need to inherit one copy of the gene from each parent. And usually the parents do not have to have the condition themselves, right? So as you can see, this makes it incredibly difficult when we're looking at phenotype. And what does phenotype mean? Phenotype means the patient in the clinic. What do they look like? How do they present to the physician? They may present and say, I don't have a family history, right? And you may not know you have a family history. So X-linked. All right, so XX, XY, right? So the mutation is on the X chromosome. So it's always expressed in males because males have one X chromosome and one Y. And then in females, it would present in those who are homozygous, right? So they would have to have those copies of the traits. Let me give you a little bit of feedback. So mitochondrial is the tricky part, right? So mitochondrial DNA mitochondria are the cells that produce energy in your body. And mitochondrial DNA always comes from the mother. So it's maternal inheritance. All right. And now switching something more clinically. So again, just kind of breaking down genetic causes of Parkinson's disease. We're still talking about Parkinson's disease. The patient looks very much the same in the clinic. They have some slowness of movement, bradykinesia, some rigidity, some stiffness of movement, and resting tremor. Maybe even some postural instability. We tend to see it more at a younger age in some of our genetic types. We still see the same pathology. When we say pathology, we mean if you did later in life when someone passes, if you did a brain autopsy, you would find that there would be changes in the brain, perhaps Lewy bodies, perhaps changes in the substantia nigra part of the brain. You'll see the substantia nigra. You'll see this slide a lot in Parkinson's lectures, but the substantia nigra there being very visible, and then somebody with Parkinson's, there's less of that red substance, right? There's less of that reddish black material, substantia nigra, right? What's in a gene? So this slide, I think it's not important for you to remember the genetic mutations that I mentioned here, but I think it's important when sometimes we want to identify patients in the office and we say, oh, they have LRRK2 Parkinson's, or they have PARK2 Parkinson's, or that sort of thing. So just seeing those are really the 5 different types that make up most of the simple mutations, LRRK2 being something you may have heard about, and that's why we name things the way we name things. So we have alpha-synuclein, we have the Parkin mutation, we have the PTEN, PINK1 mutation, PARK7, and then LRRK2. And there are many, many others, but I think some of those may be something that you might have heard about, and those are the more common ones. So moving on. My next few slides are going to sort of delve into the majority of the cases that fall into dominant, recessive, X-linked, and mitochondrial. So dominant is going to be on this slide and the next one. So autosomal dominant. So most of the names here are easy for us to remember, right? So PARK1, 3, 4, 5, and 8. All right? Over here you see the chromosome number that's involved, right? You see the gene type. And over here, again, we're highlighting the fact that the mean age of onset in a lot of these patients is young onset, right? The mean age is somewhere between 30s and early 50s. Okay, and this just stands for young onset Parkinson's disease, YOPD, young onset Parkinson's disease. You'll see some of these people have dementia, unfortunately, as part of their phenotype, again, the way they present, right, to the office. And then this over here tells us that they have a good response to levodopa, right? So I think when it comes to neurology clinics, especially a movement disorder clinic, those patients oftentimes, even though they're young, will give them levodopa. They respond really well, right? And so that really is, it's a good thing when we can find something that helps the patient. The patient's happy, the physician's happy, the family's happy. We like those that are a good response to levodopa. We know we can do things to help that patient a little bit more in their mobility. The second one, this is again the same slide, autosomal dominant, but different types. You'll see here SCA2, SCA3, FTDP17, and DYT12. Okay, now here, these types, even though they're autosomal dominant, you'll see these with a very younger age. Look here, the 23 years old is the median or the mean age of onset. These unfortunately don't respond as nicely to our treatments as we would like. right? And so some of these things, these abbreviations stand for some of the dementias. So frontotemporal dementia, right? CBD, right? Corticobasal ganglionic degeneration. So some of these things do present to the office at young ages and do have a genetic cause. They don't respond as favorably to levodopa therapy. All right, so switching colors to blue. Blue is the autosomal recessive. So these are the ones where you would need to inherit from both parents. PARC2, PARC7, PARC6, PARC7. All right, again, we're talking about younger age of onset. This time, good response to therapy. All right, and actually in this one, Parkin is linked to 77% of young onset patients under the age of 30. Okay, and even 10 to 20% of young onset in general. So we are finding genetic links, particularly with young onset people, maybe without a family history. X-linked recessive, okay? These are going to be, this is DYT3 is the one that I've highlighted here as kind of being the more common one. Dystonia, you might have heard of the word dystonia. Dystonia is sometimes a body part turning in. So sometimes you'll see an arm, a neck, toes, legs turning in on itself. Poor response to levodopa. Mitochondrial DNA, so inheriting this from the mother, right? So mitochondrial. The phenotype here, we're seeing one more with dementia at a younger age. So we're talking mean age 31, mean age 42, okay? Some are going to be a little bit more responsive to treatment than others. And when we say levodopa, that's what we study the most. It's predictable, it's easier to study, but there are many other types of treatment as well besides that in terms of medications and other treatment options. All right, so this is where I think we'll take a little step over to a clinical trial, okay? This is where I think we can advance our research and advance our knowledge in genetic subtypes. It is a completely free clinical trial. Anyone can join this, all right? And I often tell patients in the clinic, you're not involved in any sort of necessary visits or anything like that after the trial is over. It is really simply for us to gain genetic information. It's going to be stored with the Parkinson's Foundation. We're finding genetic differences in people with Parkinson's so that we can potentially offer better treatments. As you can imagine, if someone has a LRRK2 treatment, a LRRK2 mutation, and they have Parkinson's disease, wouldn't it be neat down the road to say, oh, this medicine works for LRRK2, right? This medicine we know is going to help the patient. So to date, this has been 3,000 genetic tests that have been completed by the Parkinson's Foundation. It's a free clinical trial. You can register. I'll show you how on the next slide. 14% of participants we have found to have a genetic link. So it's an information gathering clinical trial. You can register with the link. The slides I would imagine will be sent out later, or you can copy this down. You can also go directly to the Parkinson's Foundation, find it that way. You could also email genetics@parkinson.org for more information, or you can call their hotline, 1-800-4PD-INFO. What it involves, if you are interested, is you'll first have a consenting visit appointment, right? So it'll be a video visit, kind of like a Zoom appointment. You probably are familiar with doing them, or they will walk you through it. They basically want to get your consent. Is it okay that we send you this? Is it okay? This is what it would mean. This is what you have to do to get your test results. Number 2, complete a cheek swab at home, put it in the kit, mail it back. Number 3, genetic counseling appointment, right? So they're not going to just mail you a result. They're going to talk with you about the results. So I don't see many negatives in being in this study. If you're remotely interested in learning about your genetic subtype, I think it's important to call. If you have any questions, they have people working to answer the question. And when it was initially coming out, they did get a little bit bombarded with patients. And I think that they're now, the word is that they're able to fulfill phone calls pretty quickly. All right, so most people are going to be meeting criteria. So the data is anonymous, but they do share it in research. So you should know that information is going to be confidential, all right? You share with whoever you feel comfortable sharing it with. Okay, perfect. So my references are listed here, and then also the Parkinson's Foundation for that clinical trial. And this is Neuroscience Center, and then this is the Hilliard office where I see a lot of patients as well. So here's my contact information.